Is it true that there are parts of our DNA that don't actually code for anything? What are these called? What are they for? Why do we have them? In this video, we'll talk about the process of transcription and what's going on with those non-coding parts of our DNA. Let's get started. So let's revisit this idea of the central dogma of molecular biology. This is the flow of genetic information within a cell. And in the last video, we talked a lot about this process here, how DNA makes more DNA. That process is called replication. Well, in this video, we're going to focus on this step here, the process of transcription. That is when DNA is used to transcribe into messenger RNA. We're keeping the same language. We're going from nucleotides to nucleotides, but we're transcribing from DNA to messenger RNA, and you'll see that we're going to end up with a single-stranded messenger RNA there. And then in subsequent videos, we'll talk about how that messenger RNA serves to translate into proteins for the use of the cell. Okay, so we're going to focus on transcription in this video. Now let's think about this for a minute. From nucleus to cytoplasm, how does the information get from the nucleus to the cytoplasm? Because if you think about it, your DNA is locked up tight in the nucleus. So here's the nucleus, and your DNA is locked up tight in there. Now, I like to think of this, the example I use is like a recipe book. So my mother has this recipe book of generations of family recipes, and this recipe book does not leave the house, right? If you want to take a picture of a recipe, if you want to write down a recipe, that's one thing, but the recipe book itself does not leave the house. I kind of like to think of that as the DNA, right? The DNA doesn't leave the house. It doesn't leave the nucleus. So if we have DNA locked up tight in this nucleus to protect it, how do we get information to the cytoplasm where the ribosomes are going to use that information to make protein? Well, what happens, and what we'll learn in this video, is that we just make a copy, just like my mother says. Make a copy of the recipe, take a picture of it. So one recipe goes out, and you can take that recipe and you can still have the information. Well, that recipe, the individual recipe, is messenger RNA. Now, what you'll notice is that I didn't take the entire recipe book and make a copy of it. I just took the page of the recipe that I wanted to make to send out to the ribosome. So if our DNA is in the nucleus, and that's like the recipe book, it's got all the recipes to make all the proteins of your body, messenger RNA is sort of like one individual recipe within the book that is now going to go out, and you can make whatever dessert or entree from the recipe that you want. Okay, so let's talk a little bit about RNA. RNA is different than DNA in a few ways. One, it has a ribose sugar instead of a deoxyribose. And then the bases in RNA, we'll find that there's a uracil instead of a thymine. So if you're looking here at DNA, right, you see A, T, G, and C. A, T, G, and C. But notice in messenger RNA, there are no T's. We got our A, our C's, and our G's, but uracil replaces the thymine. Then later, a process we'll talk about is how translation will read the messenger RNA and turn it into the amino acids that make up protein. Okay, so without thymine, we still have our base pairing rule. C goes with G, but remember U replaced T, so U is going to bind with A. Ribosomal RNA, or excuse me, RNA, which is ribonucleic acid, is also single strand. Notice the one strand there versus DNA, you've got the two strands. So what do we have here? What are the main differences? Ribose sugar instead of a deoxyribose sugar in DNA. We've got uracil instead of thymine. And we have a single strand instead of a double strand in DNA. Now, there are many types of RNA, and that's what I'm going to go over here in the next slide. So many RNA types. I've already talked a little bit about messenger RNA. Messenger RNA is sort of like the recipe, right? It's the one single recipe from the book that's going to go out and be used to make the proteins or whatever entree we want to make, right? The protein is what makes up our body. So this messenger RNA is going to give the code to make the protein, carrying the recipe from the DNA to the ribosome, if you want to think of it as a recipe. Transfer RNA, we'll see in our next video, it essentially will transfer amino acids down to a ribosome and help make the polypeptide or protein. So it's sort of like a little adapter molecule that helps the amino acid line up with the correct messenger RNA later on. We also have ribosomal RNA, and that makes sense. It's called ribosomal RNA because it just forms the ribosome. Okay, so messenger RNA, it's like our recipe, it's our code. Transfer RNA is going to bring the amino acids down to the messenger RNA. Ribosomal RNA is what's going to help make up the ribosomes, where we're going to find this process of translation is going to occur next. Now, the last ones are a little more modern in terms of our understanding, but I want to go over a few different types because you'll probably see these in the future. snRNA has a few functions specifically in the nucleus with splicing. So snRNA, small nuclear RNA, is going to be used in some splicing of some materials that we'll see later. miRNA is what's going to be, it's kind of like a little interfering RNA, and this is going to help regulate gene expression. Right? Just because you have genes, you have the DNA, doesn't mean that DNA is turned on, doesn't mean it's expressed. So miRNA is going to help regulate if the gene is turned on or off, or if it's expressed or not. And then the last one is some other interfering RNA, and that is going to silence gene expression. So whether or not we're regulating that gene being turned off or off, if we see some siRNA, that's actually going to silence that gene and say, nope, gene, you don't need to be expressed right now. Okay? So I would say that these three on the top are the main RNA types we talk about. These three down here on the bottom are a little newer in our understanding and or a little more complex in their behavior. So let's go over the overview of transcription. So we have to start with DNA, and we've got two strands in DNA, right? So which strand serves to make the messenger RNA? Well, the strand that is transcribed is called the template strand, right? It serves as the template. The untranscribed DNA, the one that's not used, we consider the coding strand because with the exception of the uracils and thymines, it's basically the code of the messenger RNA. And I'll show you that. So what you can see here is the overview is that you've got your DNA, you've got two different strands. Notice the pink one that I've got pink up here is the template strand. That's what's going to be used to match up nucleotides to make this messenger RNA. The blue one we call the coding strand because it's basically the same code as that messenger RNA. And this enzyme here that's going to function is RNA polymerase. All right, so we got the template strand labeled for you, got the coding strand labeled, and this enzyme that's going to do it is RNA polymerase. Okay, so just like we had DNA polymerase, we've also got RNA polymerase, and this enzyme, you know it's an enzyme, it ends in ASE, is what is basically adding these new nucleotides to this growing messenger RNA strand, making the new RNA polymer. And you can see that labeled for you there, RNA polymerase. Okay, so there are really three steps to how this works, right? We've got to initiate transcription, then we've got to make the messenger RNA longer, so elongation, and then we need to terminate and stop the messenger RNA from being made. So let's start with transcription. So the initiation of transcription is when the enzyme, RNA polymerase, is going to bind to a specific place on the DNA. All right, so we got some DNA. But what gene do we want? Well, that is a very complex thing, which we'll talk about in a subsequent video. But we need RNA polymerase to come and bind to the DNA somewhere so it can start making our messenger RNA. There is a sequence on the DNA called the promoter sequence. So it's just a region of the DNA where the RNA polymerase will bind. Now remember that DNA is always read 3 to 5, so the RNA polymerase will bind to the 3' end of the template strand, and then it will go on and transcribe the gene. So if here's our promoter region, here's the actual gene we want to transcribe into messenger RNA, what we'll find is that the RNA polymerase, the enzyme, is going to bind first to the promoter region, sort of like a signal, like, hey, come over here, and then it will transcribe the gene. So there's my RNA polymerase, and I've done some fancy little animation here. You can see basically it just sort of moves down the line, and the messenger RNA will be made as it's reading. So the messenger RNA isn't coded from the promoter region. That's just where the RNA polymerase binds. Here's the gene that is transcribed into the messenger RNA. So we start with the initiation. It binds to the promoter region. And then the elongation part, right? So as RNA polymerase is moving down, right, we know that it's moving from the 3 to 5 direction. It's reading this DNA there. So as the RNA polymerase comes, it adds the appropriate nucleotide. So for an A, that's going to put a U. For an N, A, U again. For C, notice it makes a G. G, it puts a C. And it just keeps building these, right? So think about what would the next one be here. See, so the next thing in the RNA would be a G. And so this continues and continues. So we've got our DNA here. Here is our new messenger RNA that's being made. Now notice, this messenger RNA here is being manufactured from the 5 to 3' end. So even though the DNA was read 3 to 5, notice that the new strand of messenger RNA was manufactured from the 5 to 3, right? Because this is the 5' end matches this, and that's the 3' end. So it's read 3 to 5, the DNA, but the new RNA is built 5 to 3. That's called the elongation phase. Now, if you remember back to DNA replication, at the very end, DNA polymerase 1 came in and did some proofreading, did some correcting. Well, in transcription, we don't have any of that, right? No proofreading, because this messenger RNA has such a short life, it's really not worth it, and there's so many copies of it. So there's not a lot of proofreading in this process, which is why this RNA polymerase is kind of like a one-stop shop enzyme. It does all the work. All right, so now let's talk a little bit about the difference between prokaryotic and eukaryotic genes. Remember, in prokaryotes, the DNA is just in the cytoplasm because prokaryotes don't have a nucleus. Their chromosomes are circular, and their DNA, we say it's naked DNA. It's just not in a nucleus. It's kind of unprotected. And there are no introns, and we'll learn here in a moment. Introns are non-coding sequences. So over in eukaryotes, there's DNA in the nucleus, right? It's nice and protected. Our chromosomes are linear, and the DNA is wound around these histone proteins, as opposed to over there in prokaryotes, it was sort of out here in the open, not wound around these histone proteins. And in eukaryotes, we have expressed genes, or exons, and we have in-between genes, or introns, that don't code for anything. So here is a typical strand of eukaryotic DNA, right? And what you'll notice is that there's some stuff at the beginning, right? Here's an enhancer and a silencer. We talked about this earlier. Does this gene need to be turned on or off? These enhancers and silencers will help with that. They're kind of downstream from the actual gene. This is the gene that we're really going to read. We call it the ORF, the open reading frame, right? That's the gene to read. That's the gene that needs to be transcribed into messenger RNA. Then there's a promoter region, right? This is where the RNA polymerase will bind. Here's the gene that's going to be read. It will eventually get to the terminator after it's done reading and stop. There's also some enhancers and silencers maybe controlled down distal from the original gene. But I want you to notice these two things. Notice this gene to transcribe has some introns and some exons. The introns are non-coding. They don't code for anything, right? That's what we started the whole video with. Yes, in your DNA, there's part of that DNA, some nucleotides that actually don't code for anything. Now, why are they there? Probably something left over from evolution we don't use anymore. Or it could be for several other reasons. Maybe we just don't understand what they're there for. But these, as we know, don't code for anything. The exons, however, these little darker blue, these do code for something, and we say that they're expressed. So the exons are expressed sequences in our DNA, okay? So just as sort of a general overview of this again, eukaryotic DNA has a region way down that will turn on or off the gene. Then there's a place where the promoter, where the RNA polymerase will bind. Then there's exons and introns in the actual gene that's being transcribed. The exons code for things, the introns do not. And finally, there's a terminator region which tells the RNA polymerase to stop. Okay, so after this is all done, after the DNA is converted to messenger RNA, a few things need to happen. We call this post-transcriptional processing. So after transcription, post-transcription, we need to do a few things. That eukaryotic messenger RNA needs some work, right? Think about this. It's been nice and protected in that nucleus. But what we need to do is, if it's going to go out into that scary cytoplasm to find the ribosome, it needs to be protected a little bit. So here's our pre-messenger RNA, right? This is what's been made. Well, to protect the messenger RNA, we do a few things. One, we add a 5' cap to the 5' end. We also are going to add a poly A tail. So notice this. So if this is our pre-messenger RNA, on one end, we're adding a 5' cap. That would be the 5' end. And then on the other, we're adding a 3' poly A tail. This is just a series of adenines that repeat over and over. And basically what these do is they sort of bookend the messenger RNA. In other words, this is a protective mechanism. So some other enzyme won't come and sort of eat it. If it does, it's fine because here's the real important gene in the middle. But this protection on the ends helps keep it safe to go over to the ribosome. The other thing we got to do is, before we send it out of the nucleus to the ribosome, we want to edit out the introns. Remember, the introns are non-coding. So we're going to take out these non-coding sequences, which will just leave the exons, which are expressed as the spliced and processed messenger RNA, which is now ready to go out into the nucleus to find the ribosome. Okay, so just a review of what we talked about in this video. This video was all about going from DNA to the messenger RNA. And that process is like taking one recipe from the recipe book and making a copy of it so we can send that recipe off to the chef, the ribosome, who is going to make us the nice dessert or meal that we need or make our proteins for us. That will come in the next video. So my next video is on translation, and that will tell you and explain the process of how this messenger RNA is used to code and is translated into protein.